Hello, Team IABC 10.
Hello.
Hello.
Good morning to you.
Welcome to the Global Youth Science and Technology Bowl 2020. Team IABC 10 is from Ireland. Their project title is an R&D study on using a nutrient treatment biofilter to combat oceanic and freshwater dead zones, which harnesses the intrinsic potential of halophyte species. We would like to thank you for attending the online judging today and for your efforts made in the past few months. Today, we are happy to have Professor Lam Hong Wah from City University of Hong Kong, Dr. Wong Chat Loi Carmen from the University of Hong Kong, and Dr. Wong Yi Kern from Episteme Co. Limited to be the judges of the competition. You will have 5 minutes to present your project. When time is up, we will stop your presentation. After that will be a 4-minute Q&A session. You will be able to see a timer on my screen. Judges, are you ready? Team, are you ready?
Yeah.
So you can please share your screen now.
Okay.
Okay, you can start your presentation.
Yeah. So my name is Timothy McGrath. I'm from Killorglin in County Kerry. So the motivation for my project came back in February 2019 when I learned of Boyan Slate's work with the Ocean Cleanup Project. So my research advanced and I found eutrophication was a big problem in a lake near where I live. And on further observation, eutrophication, I realized turned into areas called dead zones, which heavily impact the Irish coastline and other places around the world, such as the East China Sea. So our world has a water deoxygenation crisis in our hands, and we aren't solving the societal challenge. So the existing knowledge that water bodies start out classified as eutrophic, meaning they have excessive nutrients such as nitrogen and phosphorus from fertilizer and sewage discharge. This induces excessive growth of algae blooms, and this then causes areas of hypoxic, and then this persists and forms areas called dead zones. So the hypothesis for my project was by using botany and engineering to use native halophyte plants to decrease the amount of nutrients in the water, solving the serious problem of dead zones by ceasing the growth of algae. Furthermore, this could then be used to produce bioenergy and making the project more commercially viable. So that's an overview of the projects or experiments that I went through. So the first experiment was a quantitative and qualitative study. This was to find suitable halophyte plants that I could use in my project. So I performed these studies and also on the aquatic factors of a seashore, which is classified as eutrophic. So I analyzed soil salinity, root and shoot system dimensions of the different halophytes. And my results came that I found 2 plants, Spartina anglica and Salicornia europaea, as 2 halophytes that could withstand the salt tolerance of the estuary water. The second experiment was to investigate abiotic factors, and this was to actually get more research for the actual next experiment, which was using these data that I collected for the dead zone conditions in my school laboratory. So I tested the 2 plants under dead zone conditions that I had from my primary research and secondary research. And I found that Spartina anglica had a greater uptake of nitrogen. So it was nitrate, and I continued my research with that. So then I actually created a prototype of my system, which you can see there in the images. And I assessed the flotation and function of the system out in an estuary where I live under calm conditions, and it was very successful. I continued my project and did the assessment of a small scale flotation system, which is the model that I have right here. And that was to see how it could stand the actual impact of waves and different conditions. I then did assessment of the system in an inland aquatic ecosystem, as the project can also be used for fresh water. So I also then actually tested the temperature of the plants and how they can sustain under the temperatures of the system. The actual building the system, I use CAD software and SolidWorks to actually develop it and create it myself, but in the future it can be created more sustainably and more into building it for manufacturing. The further research, I contacted different institutes such as UCC here in Ireland and the Marine Environmental Institute here. And they saw my project as a fantastic and interesting novel solution, and that it could actually be used for integrated multi-trophic aquaculture systems or at factory discharges as well. It's also has been seen that my project will be a very good use for bioenergy, and I continue to research that into the use of circular economy, which means that the plants could then be used as for bioreacting and actually create energy out of it. So this is the presentation backboard that I have behind me. So it gives an overview of the actual project that I have done. And thank you for listening. Thanks.
Okay, the